Our major research interests relate to what is referred to as in vivo gene therapy. That is, using genetic material as a drug and transferring that directly to individuals with disease.
Whatever you see on the microscope with a blue color, those are cells expressing a new gene after in vivo gene transfer.
Patents go, your sales go down, you've got this huge manufacturing, distribution, sales, and marketing organization, and you have to feed it with products. I think gene therapy offers the greatest potential of anything I've seen in the industry. And I'm convinced that in 15 or 20 years, most of our products will be delivered DNA.
The gene therapy field has really exploded over the last 3 years. Currently, there are approximately 40 to 50 gene therapy companies in the United States.
We now have the ability, essentially, to transfer genes to any organ of the body. We can do it either by taking cells out of the body, such as blood cells, and putting them back in after we modify them, or by modifying them, as we're doing, directly in the body, which we can essentially do for almost any organ now.
At this point, over 200 patients have been treated with gene transfer products in the United States. And by the end of this year, I expect that that number will close to double.
Biotechnology industry is interested in trying to develop lines of mice or perhaps eventually other animal species which will act as sort of factories for producing large quantities of a particular human protein that at the moment by existing techniques are very difficult to produce. Having derived clones that contain an altered or mutated gene that we're particularly interested in, we then want to put those cells carrying that mutated gene back into the embryo so they can, in a sense, participate again in their normal developmental program.
One of the issues that I believe is going to become an important issue in gene therapy is the issue of germline transfer. The notion of having a gene get into a sperm cell or an egg cell and then having that passed on to a future generation, even accidentally, just has tremendous humankind implications.
It has moved much faster than anybody would have dreamed 5 or 6 years ago. We haven't had any problems yet. I mean, there are going to be problems. It's inevitable when you're developing a new technology. But I think the promise is just extraordinary.
So we're dealing here with the technology of using embryonic stem cells to genetically alter the genes or particular genes in the mouse. With a very fine glass pipette that contains the injected embryos, we put that into the uterus and expel the embryos. It's almost exactly identical to the same technique of transfer of fertilized eggs and human test tube babies. And the female usually recovers within about 15 to 20 minutes after this operation.
There's no question that what's happened in molecular biology is truly revolutionary in terms of medicine. And I think if it's open and disclosed, I don't think there are significant concerns for society.